Oh, hi, Alyse. Maybe we'll give just like a minute or two further to see if there's any late joiners, and then we'll start.
Yeah, sounds good.
Okay. Okay. I think it looks like we picked up a couple more people, so I think we're good. Welcome to everybody. Thank you for taking the time to listen to the seminar series. Today, Alyse Staley is going to present on the BOIN design. And she's a biostatistician with the Bioinformatics and Biostatistics Shared Resource. So I'll just turn it over to Alyse, and looking forward to this presentation.
Great, thanks for the introduction, Andrew. So as Andrew said, today I'll be presenting the Bayesian Optimal Interval Design, or BOIN. And this is a phase 1 clinical trial design that our core highly recommends because it's effective, flexible, and transparent. Today, I'll first start by giving a general overview of phase 1 clinical trials, including their objectives and commonly used designs. I'll then delve into the BOIN design specifically, including a comparison of BOIN versus the commonly used 3+3 design. Then I'll go into BOIN in more depth, which will include the inputs, what we as biostatisticians need from investigators, how the design works, and finally the outputs, how it's implemented. From there, I'll go through an example of the BOIN design in practice. We'll briefly introduce some extensions of BOIN and end with a brief summary. When a new drug is developed, it first goes through non-human or preclinical research. This can include many methods such as computer simulations and animal models. Once the drug is ready to be tested in people, it's tested in 4 distinct clinical trial phases. There are many overlapping secondary and exploratory objectives for each phase, but each phase's primary objective is clear and distinct. Phase 1, which will be the focus of today's lecture, is used to assess safety and the proper dose of a new drug. This is the smallest phase clinical trial and generally has somewhere between 15 and 50 patients. Although for simple phase 1 designs in cancer applications, we generally see on the lower end closer to 15 patients. If a drug is deemed to be safe, it can then move on to phase 2, which assesses its effectiveness. And this is a slightly larger trial. Then if the drug is deemed to be both safe and effective, it can go on to phase 3, which compares the new treatment to existing treatments. This is the largest phase and is generally in the format of a randomized controlled trial. Finally, once the drug is FDA approved and is being prescribed by physicians, it then enters phase 4, where the long-term side effects are observed in the general population. Again, the primary objective for phase 1 is safety. Specifically, we have to ask 2 questions. 1, is the drug safe enough to proceed to phase 2? If not, we need to terminate the drug trial and the drug does not move on to phase 2. If it is deemed to be safe, the next question we have to answer is at which dose should patients be treated at in phase 2? For cancer applications, it's generally accepted that the higher the dose patients are treated at, the more effective that treatment will be. More formally, this is called a monotonicity assumption. Therefore, we want to find the highest dose that patients can tolerate. More specifically, we want to find the maximum tolerated dose, or MTD, where the MTD is defined as the dose with the DLT probability closest to the target DLT rate, where a DLT is a dose-limiting toxicity, or a toxicity that is deemed unsafe enough that a patient needs to stop taking the medication. For example, if we're trying to have a target DLT rate of about a third of patients experiencing a dose-limiting toxicity, we want to find the dose that gets as close to having a third of a patient experience a DLT as possible. There are three main categories of phase 1 clinical trial designs. First, there are rule-based designs, including the commonly used 3+3 design. I will talk about shortly, the 3+3 design is straightforward to implement, but it performs pretty poorly. And so it's generally not recommended, although it is still commonly used. Then there are model-based designs, which perform quite well and are very flexible. However, they're complex to implement, so they've been met with a lot of resistance from PIs. And finally, there's the model-assisted designs, which include BOIN, the topic of today's lecture. And these take the advantages of both rule-based and model-based designs, and you end up with a well-performing and flexible design that's also simple and transparent to implement. I'll now briefly go through how each of these types of designs work. For the rule-based design, I'm going to present the 3+3 design since it's most commonly used. And this design has poor precision for choosing the MTD, and it's not very flexible. So patients have to be enrolled in cohorts of 3, and the target DLT rate is set at 30%. In short, the design works by first enrolling 3 patients at the lowest dose. If of those 3 patients, none of them experience a DLT, then the next 3 patients will be escalated to the next higher dose. If 1 out of 3 patients experiences a DLT, then 3 additional patients will be enrolled at the same dose, and we'll use the information from all 6 patients to decide whether to escalate the next cohort of patients or to stop the trial. In the case where the first 2 out of 3 patients experience dose-limiting toxicities, the trial is terminated. And this repeats with all of the cohorts in the trial. Once the trial is determined to stop, so when you're having 2 out of 3 DLT, then the MTD is chosen as the prior dose. In other words, if dose level 3 required the trial to stop, dose level 2 would be our MTD. For the model-based designs, I'll present the continual reassessment method, or CRM, because again, it's one of the more commonly used model-based designs. This design is well-performing because it accurately targets the MTD, and it's flexible because it allows for a customized target DLT rate. Its drawback is that it requires real-time biostatistical support to make each dose escalation or de-escalation decision. The CRM works by first using a statistical model to model the dose toxicity relationship based on prior knowledge. The first patient is then assigned to the dose that is close to the current estimate of the MTD based on the model. The model is then updated based on the first patient's observed outcome, and the next patient is rolled at an updated estimate of the maximum tolerated dose, or MTD. This process keeps repeating after each patient until all information from all previously treated patients have been used. So you're updating this continuously and never throwing out any information. Finally, there are the model-assisted designs that include BOIN, as well as some alternatives that work pretty similarly. So that's the keyboard MTPI design, as well as MTPI2. Like model-based designs, statistical models are used to determine the optimal escalation and de-escalation boundaries for model-assisted designs. And also like model-based designs, there's a customized target DLT rate, and the cohort size can also be customized. Like rule-based designs, these escalation rules are provided to investigators ahead of time in a table. That way, investigators know exactly when to escalate or de-escalate and when to stop the trial. I'll go into this in a lot more depth when we talk about the BOIN design specifically. To summarize, the rule-based design is simple and transparent because the physician knows at which dose the patient should be treated based on a pre-specified table of escalation rules. However, it's inflexible and doesn't perform well. The model-based design is very well performing, but it's considered both statistically and computationally complex. So it's been met with resistance from PIs due to its closed box type approach. And finally, there's the model-assisted designs, including BOIN, that take the advantages of both the rule-based and model-based designs while eliminating many of their shortcomings. BOIN is implemented very similarly to the 3+3 design while allowing for a lot more flexibility and higher precision. Now I will delve into the BOIN design specifically. The Bayesian optimal interval design is the most commonly used model-assisted design, and it works by comparing the observed dose-limiting toxicity, DLT, rate with a pair of fixed and pre-specified dose escalation and de-escalation boundaries. Generally, the first cohort of patients are treated at the lowest dose level. If the observed DLT rate is less than the pre-specified escalation boundary, so you're in this blue area, then the next cohort of patients will be treated at the next higher dose. If the observed DLT rate is within the escalation and de-escalation bounds in this stay area, then the next cohort of patients will be treated at the current dose. And finally, if you're in this red area where your observed DLT rate is greater than or equal to your de-escalation boundary, the next cohort of patients will be de-escalated. This is repeated until either the maximum sample size is reached or the trial has to be terminated due to excess toxicity. If you aren't already convinced of the benefits of BOIN over the rule-based 3+3 designs, I'm now going to delve into more detailed comparisons between these two designs. First, BOIN has an advantage over 3+3 due to its flexibility. The 3+3 design has a fixed DLT rate that's set at 30%, whereas the BOIN design allows this DLT rate to be flexible. This is beneficial for certain applications. For example, there might be a population of cancer patients that don't yet have an effective treatment. Therefore, a DLT rate greater than 30% might be desirable. Additionally, the 3+3 design has a set cohort size of 3, whereas the BOIN allows this to be customized. This is helpful perhaps if an investigator wants to use titration, where they might start with a cohort size of 1 and adaptively increase that to a larger cohort size. This might be used when there is a very small population of eligible patients and there's strong evidence that the first dose is very safe. The BOIN design also allows de-escalation doses, so you don't necessarily have to start at the lowest possible dose. And finally, it allows you to recruit patients even when you've deviated from the original plan. For example, if a patient is unable to finish the study, you could have a cohort of 2, even if you originally designed it to have a cohort of 3, without throwing out any information. BOIN is also superior in terms of its performance. First, The 3+3 design calculates the maximum tolerated dose as one dose lower than the dose that got at least 2 DLTs. So if dose level 3 had 2 patients with DLTs, then dose level 2 would be your MTD. And this isn't based on much information. In contrast, the BOIN design pools information across all cohorts to get a more informed decision of the MTD. Additionally, the 3+3 design has an excessively small and random sample size. Therefore, no more than 6 patients can ever be treated at any dose, which just isn't much information to make these decisions on. For example, you might observe a DLT rate of 16.7%, or 1 out of 6 patients experiencing a DLT. And that is really low. But due to such a small sample size, if you look at the upper bound of the confidence interval, the true toxicity rate could actually be as high as 64%. If you have a high observed DLT rate of 50%, your true toxicity rate could also be as low as 12%. The BOIN design, in contrast, uses a more appropriate sample size that then doesn't require post-hoc expansion. Next, the 3+3 design is overly conservative in terms of dosing, which means it has poor precision in identifying the MTD. At first, conservatism in this context may seem advantageous. However, misidentification of the MTD has serious consequences when you both over and underdose. Of course, if you treat patients at an overly toxic dose, that's dangerous. But if you treat patients at subtherapeutic doses, that means that in phase 2, when you're studying efficacy, you might overlook a treatment that could have been really effective at a higher dose. And this is also a waste of resources. BOIN, on the other hand, has a much better balance between safety, so your risk of overdosing, and identifying the MTD. I'll now go into the BOIN design in more depth. First, we as biostatisticians need information from the investigator to design the phase 1 clinical trial. First, we need to know the maximum number of participants that are expected to be enrolled, the desired cohort size, and any prior knowledge about safety. The BOIN design has many built-in safety rules that are sufficient in the vast majority of cases. But if an investigator is abnormally concerned about safety, there are actions we can take within the BOIN design, such as implementing a stricter stopping rule. Conversely, if there's strong prior evidence that the first dose is safe, we can alter the BOIN design to stop the trial early if it converges to a given dose. Additionally, we need to know the target dose-limiting toxicity rate, or DLT. This is often set at 0.3, but some applications it may be appropriate to deviate from that. In addition to the target DLT rate, we also need to know the range of acceptable DLT rate. This is because there's a small sample sizes, so the true DLT rate might be a bit higher or lower than our target rate. Therefore, we need to first know Phi1, which is the highest DLT rate deemed underdosing. In other words, if a certain rate is observed, when does escalation have to occur? And Phi2, what's the lowest DLT rate deemed overdosing? In other words, at what DLT rate does de-escalation have to be required? By default, the acceptable range of DLT rates is calculated by multiplying the DLT rate, or the target DLT rate, by 0.6 and pi 1.4. For example, at our target DLT rate of 0.3, we could calculate our Phi1 value as 0.6 times 0.3 equals 0.18, and similarly, Phi2 equals 0.42 by multiplying 1.4 times 0.3. In addition to these default ranges, you can also customize the DLT acceptability range. In most cases, the default is highly recommended, but it is possible to use slightly narrower ranges. For example, we could use a more conservative de-escalation rule, and instead of using a coefficient of 1.4, we could use a coefficient of 1.2. However, you have to be really careful with this, because if we make the boundaries too narrow, there isn't enough power to distinguish between the escalation decisions. So it's actually recommended to alter the target DLT rate itself in order to achieve an acceptable range. For example, if we brought 0.3 down to 0.25, we'd be multiplying phi2, 1.4 times 0.25, and that would also bring down our de-escalation requirement. Once we have all this information from investigators, we then need to calculate the escalation and de-escalation boundaries for a given dose. The idea behind this is to minimize the incorrect decisions of dose escalation and de-escalation by calculating optimal escalation and de-escalation boundaries. Continuing with our previous example, we have the target DLT rate at 0.3 and our acceptable range of DLT rates between 0.18 and 0.42. We plug these values into these formulas, and then we get our boundaries. So we have 0.24 and 0.36. This is saying that if more than 35.9% of patients experience a DLT at a given dose level, we'll de-escalate. If you're within these bounds, we would stay the same. And if more than 23.6% of patients had, if less than 23.6% of patients experience a DLT, then we would de-escalate. Additionally, the BOIN design has built-in safety rules, such that if a dose is too toxic, it can be eliminated entirely from the trial. This occurs if two criteria are met. First, we want to calculate the probability that the observed DLT rate is greater than the target DLT rate. If that probability is greater than 0.95, our first part of the criteria is met. The second part of this is we must have at least 3 patients who are treated at the current dose or higher. We don't want to make any decisions based on only 1 or 2 patients. If both of these criteria are met, then the current dose and any higher doses are eliminated. If the lowest dose is eliminated, the trial is terminated. Now we're ready to implement the trial that we designed. And this follows the following flow diagram. We generally start at the lowest dose or whatever the pre-specified starting dose is. We then go through this cohort where we're treating 1 cohort of patients, and we must ask if the maximum sample size has been reached. Since this is our first cohort of patients, it has not. So it's now time to compute the dose-limiting toxicity rate at the current dose. As a reminder, the DLT rate is calculated as the total number of patients who have experienced a DLT at the current dose divided by the total number of evaluable patients treated at that current dose. It's really important to emphasize here that this is all patients treated at the current dose, not just this cohort. And this is a key difference between the BOIN and 3+3 design that contributes to its superior performance. Once the DLT rate is calculated, we then have to decide whether to escalate, retain, or de-escalate. If our DLT rate is less than or equal to 23.6%, we're going to escalate the dose. If it's greater than 35.9%, we're going to de-escalate the dose for the next cohort of patients. And finally, if our DLT rate is between these boundaries, we will enroll the next cohort of patients at the current dose, and we will go through this flow diagram over and over until either the maximum sample size is reached or the trial is terminated due to excess safety concerns. A table is also provided so that investigators don't have to calculate that DLT rate by hand for each cohort. In this table, across the top, we have the number of evaluable patients treated at the current dose. Then in the body of the table, we have the number of observed dose-limiting toxicity. And finally, over on actions, based on the number of DLTs, we need to know whether to escalate, de-escalate, or eliminate the dose entirely. For example, if 6 patients have been treated at a given dose so far, and one of them has experienced a DLT, then the next cohort of patients will be escalated to the next dose. You can see there's a couple NAs here, and that's simply because we can't eliminate the dose if we don't have at least 3 patients currently treated at it. Generally, the table is pretty straightforward to follow, but there's some cases where it's helpful to refer to a set of table rules. When the table indicates elimination, that means the current dose needs to be removed from the trial as well as all higher doses. And when a dose is eliminated, the next cohort of patients must be de-escalated to the next lower level. But if the lowest dose is eliminated, the trial needs to be stopped. Additionally, there are some DLTs that are not on the table, and that's because they're in between escalation and de-escalation, and therefore the next cohort of patients need to stay at the current dose. There are also cases where it's a little unclear what to do because you're already at the lowest or highest dose that the trial is exploring. If the current dose is the lowest dose and the rule indicates de-escalation, then the next cohort of patients will continue to be treated at that lowest dose unless the number of DLTs has reached that elimination boundary. In the case where you're already treating patients at the highest dose and the rules indicate escalation, then you will continue to treat patients at the highest dose. Once the trial has been completed, it's time to choose the maximum tolerated dose, or MTD, which is entirely separate from the escalation and de-escalation rule portion. The MTD is selected by computing a non-parametric regression or isotonic regression estimates of the toxicity probabilities of each dose. We want to choose the dose that has the estimated DLT rate that is closest to the target DLT rate. And in the case of ties, the highest dose is selected as long as it's still lower than the target DLT rate. This is a bit clearer visually. Here's an example where there are 5 dose levels of interest and the target toxicity rate is set at 0.3. Here are the estimated DLT rates for each dose level. Dose level 1 and 2 have very low DLT rates, and then there's dose level 3 that has an estimated DLT rate of 0.27, which is very close to our target of 0.3. Dose level 4, that's pretty high, and dose level 5, which the trial never reached, and so we have no information on. This is also shown in the plot here, and it makes sense that dose level 3 is chosen as the MTD, because if you look at dose level 1 and 2, the confidence intervals don't even cross our target DLT rate. So the DLT rate is much too low. Whereas dose level 3, our DLT rate is just below our target, and the confidence interval expands above and below our target. Dose level 4 is too toxic, and we haven't assessed dose level 5. To make this clearer, I'll now go through an example of how to implement the BOIN design from start to finish. Let's say we have a new drug that is being used to treat a subpopulation of cancer treatments without an effective treatment. Therefore, the investigators would like to set the target DLT rate to 0.35. The next step is to make sure the toxicity ranges are acceptable. We'll use the default using our constants of 0.6 and 1.4 to get 0.21 and 0.49, which are a bit higher than we had discussed earlier because our target DLT rate is now 0.35 as opposed to 0.3. Since this is a standard example, we'll say that the investigators are fine with these toxicity ranges. Additionally, we find out from the investigators that the maximum number of patients they expect to be able to recruit is 18. Cohort sizes of 3 are acceptable, and they're interested in testing 3 separate doses, with 1 being the lowest dose and 3 being the highest dose. Now, to implement the BOIN design, we have to calculate the optimal escalation and de-escalation boundaries based on the acceptable DLT ranges. They have been calculated using a formula here, such that we need to escalate at the 28% mark and de-escalate if our DLT rate is greater than 42%. And in this case, we'll use the default safety rules, as is done in the vast majority of applications. We'll then follow the same flowchart where we start with dose level 1 and continue through this, calculating the DLT rate each time until we've either reached the maximum sample size or the trial has to be terminated due to excess toxicity. On the top in blue is the escalation and de-escalation rules table. Let's say we first treat a cohort of size 3 at dose level 1, and we observe 0 dose-limiting toxicities. We can now refer to our table and navigate to 3, because we've treated 3 patients, and find the number of DLTs on the table. In this case, there were 0 DLTs. And we can now go over and see what the rule is. So we need to escalate for the next cohort of patients. The next cohort of patients is treated at dose level 2. And we again are able to enroll a cohort size of 3. And of those 3 patients, 1 patient experiences a DLT. Since this is the first cohort of patients treated at dose level 2, our total number of treated patients at dose 2 is also 3, and the total number of DLTs at dose 2 is 1. We'll again go then to 3 patients, and since 1 is not on the table and it's in between 0 and 2, that means we're going to retain, meaning the next cohort of patients is also going to be enrolled at dose level 2. Again, we'll assume we're able to enroll 3 patients in this cohort. And let's say none of them experienced a dose-limiting toxicity. This time, since this is our second cohort of patients treated at dose level 2, the total number treated is 6, and across both cohorts, the total number of DLTs is 1. This time we'll navigate to 6, 1 observed DLT, which means we need to escalate. And now we're up to dose level 3, cohort size is 3, 2 DLTs. Since this is the first time treating at dose level 2, those are the same as our totals. And so we'll go back to 3, we had 2 DLTs, and we need to de-escalate. And we're back at dose level 2 again. Again, we have a cohort size of 3, and let's say this time we've had 2 DLTs, and when we add up all 3 cohorts, we had 9 total patients treated, and if we add up all of the DLTs at this dose level, we have 3 DLTs. And again, we'll go to our table, this time till 9. 3 is in between our escalation and de-escalation, so we will retain. We're now getting to the end of the trial, and we know we have to enroll more patients at dose level 2, but even though we expected 18 patients to be enrolled, we actually only have 2 remaining patients available. And that's not a problem with the BOIN design. So we can now enroll a cohort of 2, and one of them experienced a DLT. The trial is now complete because we have reached our maximum sample size of 17 patients. It is now time to select the maximum tolerated dose based on the number treated and the number of DLTs at each dose level. In this case, again, our target DLT rate is 0.35, and the MTD based on isotonic regression was selected as dose level 2. And this makes sense because if we go over to dose level 2 and estimate the DLT rate, it's set at 0.36, which is very close to our target of 0.35, which is also clear from the plot. There is a wide range due to small sample sizes, so here's our confidence interval, and we believe the chance that the true toxicity rate is greater than our target is about 50%, which again makes complete sense by the confidence interval. Now we have gone through everything that we need to know for a general or simple BOIN design trial, although there are some extensions for special cases. The first of these extensions addresses late onset toxicity. In some cases, DLTs may occur long after patients are initially exposed to a drug. This means that while we are waiting for those long periods of observing dose-limiting toxicities, there are patients who are waiting to be enrolled who could have been benefiting from the treatment. But we don't know at which dose to treat these patients because the toxicity data from previous cohorts is still pending. The goal of the late onset design is to provide real-time dose assignment for new patients while that toxicity data are still pending for some of the enrolled patients. This has the benefit as well of shortening the trial duration. Additionally, there's an extension called the optimal biological dose, which combines phase 1 and phase 2. In general, phase 1 evaluates safety and phase 2 evaluates efficacy. However, this assumes monotonicity, or as the drug becomes more toxic, it also becomes more effective. In cases where this isn't believed to be true, we need to simultaneously evaluate both toxicity and efficacy in the same phase. Finally, there is the combination extension. Due to time constraints, this is the only extension I am going to present in more depth. As cancer treatments have improved and developed over time, investigators are often interested in combining multiple drugs. It's often the case where both of the 2 drugs have been FDA approved, but the interactions between those drugs haven't been studied yet. So this design finds the best combination of 2 drugs when each of the drugs has multiple doses of interest. For example, let's say we have 2 drugs. Drug A, which has 3 doses, A1 being the lowest, A2 being in the middle, and A3 being the highest. And drug B has 4 doses, ranging from B1 to B4. At first, we know that the first dose we should be treating patients at is the lowest dose, which is clearly A1, B1. But the statistical challenge comes when we have to decide which dose should be escalated to next. For example, should we escalate to A1 or B2? How about A2 or B1? And furthermore, as we get further in the trial and really don't yet know the interactions, is it possible that A1 and B4 could actually have a lower DLT rate than A2 and B2? We have to complete an optimization problem to answer these questions. Simply, the optimization problem can be thought of like an indifference curve, which you might have been exposed to if you've ever taken a microeconomics class. In this example, we have Reese's on the x-axis and Kit Kats on the y-axis. I go to the grocery store, I'm going to pay with cash, and I have a limited amount of cash on me. I know I would like to buy a combination of Kit Kats and Reese's within the constraint of my budget. And although I want both Kit Kats and Reese's, I am indifferent to the exact combination. For example, I would be equally satisfied or indifferent with 4 Kit Kats and 2 Reese's, as I would be if I had 2 Kit Kats and 5 Reese's. Anywhere along this indifference curve, I am indifferent or equally satisfied. Yet as I move further away from the origin, I am jumping onto better indifference curves, if you will, where I'll be getting even more enjoyment or utility because I could have enough money in this case to actually buy 5 Kit Kats and 5 Reese's. Bringing it back to drug trials, the concept of MTD contours is very similar to the concept of indifference curve. A contour consists of multiple combinations of drug A and drug B that have the same estimated DLT rate. For example, on this contour, we have a DLT rate that's estimated to be 0.4. And this can be achieved through a high dose of drug B and a low dose of drug A. And it can also be achieved with a high dose of drug A and a low dose of drug B, or perhaps somewhere in the middle. In phase 1, we want to identify an MTD contour with the DLT rate closest to our target DLT rate. We want to select which contour we're going to identify. Then in phase 2, that's when we want to explore which combination within the same contour is most effective. And so to identify the MTD contour, we can use the extension of BOIN called the BOIN Waterfall design. This divides the optimization problem into subtrials, where each subtrial can be thought of as its own BOIN design problem. So for each subtrial, we have escalation and de-escalation rules, and we're going to follow the same procedure that we already presented at the earlier parts of this lecture. Each subtrial identifies one candidate MTD, and then we build an MTD contour that is made up of the MTD candidates from each of the subtrials. Here is an overview of the waterfall design. We have drug A on the y-axis and drug B on the x-axis. And we have each subtrial, so subtrial A, B, C, and putting it all together in D. We're going to start by performing subtrial A. Since this is the first subtrial, we need to start at the lowest dose, which is A1B1. Then we go through the BOIN escalation and de-escalation rules just as before, and we follow the order of escalation dictated by this subtrial. In this case, A1B1 is our starting dose, and we're going to escalate A first. So if the BOIN design tells us to escalate, we'll go up to A2B1. If we escalate again, it will go to A3B1 and then start escalating along B. Let's say we performed this entire trial and we ended up with an MTD candidate of A3B2. It is now time to start subtrial B. We use the MTD candidate from the previous subtrial to determine the starting dose for the second subtrial. Before we had A3B2, now we're going to subtract 1 from A and add 1 to B. That way we are going to use a starting dose of A2B3, and we'll again perform the BOIN procedure. This time we have some available starting dose, escalation dose, and de-escalation dose. Other doses are not available to de-escalate to because they've already been tested in the first subtrial. Let's say we run through this whole subtrial and our MTD candidate is A2B3. We'll now move on to our last subtrial and similarly calculate the starting dose of subtrial C by subtracting 1 dose level from A and adding 1 dose level to B for a starting dose of A1B4. Since this is our highest level of B, we only have 2 de-escalation doses available. And we will say in this case that A1B4 was the MTD that was identified. Now we can put it all together, and you can see in D is a summary of each one of our MTD candidates, which makes a contour. At this point, phase 1 is complete, and as this was moved on to phase 2, the goal of phase 2 would be to determine whether any of these candidate dose combinations was effective, and if so, which one was most effective. In summary, the model-assisted BOIN design is both transparent and effective. That's because it uses the advantages from both the model-based and rule-based designs while mitigating some of the shortcomings of these two designs. It is highly flexible because it allows you to choose your own target DLT rate and cohort size that can be adapted throughout the trial. Additionally, there are many extensions of BOIN that are available for more complex designs. It is therefore a phase 1 clinical trial design that our core highly recommends. Today's talk was put on by the Biostatistics and Bioinformatics Shared Resource, and our Biostatistics core can help you with any kind of biostatistical analysis, manuscript assistance, or study design, including the BOIN design. We're happy to help you with any of your projects, so please reach out to us at our shared email here if you have any projects of interest. I've also included some select resources from today's talk, and this lecture will be recorded and uploaded to YouTube if you want to reference it later. With that, I conclude today's talk, and I'll open it up to any questions. Thanks. All right, well, if there aren't any questions, thank you all for coming.
Awesome. Thank you very much, Alyse. And once again, thank you everybody for joining us. Look forward to seeing you again in the future.